present on phospholipid scramblases, and it's by Ashley McAleese and Erica South. So just some basics on phospholipid scramblases. They are enzymatic proteins that are responsible for the translocation of phospholipids in a lipid bilayer. And they're found in a lot of different type of eukaryotic organisms, from fungi, invertebrates, mammals, humans. There's many different variations and shapes, and even in the same families, and the variations in shape also lead to a different amount of functions that these protein families are capable of. The two families I'll be speaking about will be the PLCR family and the TMEM16 family. So here is a diagram of a phospholipid scramblase that's in the TMEM16 family. It's the TMEM16F phospholipid scramblase, which are transmembrane proteins, so it's embedded in the bilayer. And you have the phospholipids being inverted, and then the blue shows the calcium ion channels that this type of protein also conveys. So. Okay, so for the structure of phospholipid scramblases, they basically depend on calcium concentrations. Calcium is high, scramblase activity is high. When it's low, scramblase activity is low. In order for calcium to bind to phospholipid scramblases, they have to have an EF hand domain, which is typically the calcium binding site. And this is just a helix loop helix structure. A lot of them can contain NLS regions, which are nuclear localization signal regions, which is important for nuclear transport. And that is due to important binding. It needs to be able to bind to the protein in order to move it to the nucleus. And then these are not type ATP dependent proteins, so they don't need ATP in order for catalysis to occur. And then there are many different shapes, again, lead to many different functions. The phospholipids asymmetry in the plasma membrane leads to phosphatidylserine externalization, which is a type of glycerophospholipid, and it acts as a signaling molecule when it is externalized outside of the bilayer. And this is crucial for a lot of different pathways, such as apoptotic pathways and blood coagulation cascades. So here, it just shows two different of the pathways that I just mentioned. So on the left here, there is the blood coagulation pathway. When you have presence of intracellular calcium, there is the externalization of the phosphatidylserine, which causes signaling for prothrombin, like procaspase type complexes, and basically they bind to the outside of the external layer of the bilayer, and that causes the production of thrombin, which ends up leading to different activities that cause blood coagulation. And then here is the apoptotic pathway. It just basically shows the externalization of the phosphatidylserine causes signaling for macrophage activity, which engulfs the apoptotic bodies when the cell fragments. So the phosphatidylserine actually externalizes as apoptosis is occurring in the cell, and then macrophages just phagocytotically engulf the cell.
So a study that we ended up looking at that had to do with phospholipid scramblase was looking at its regulation through protein kinase C delta in apoptosis and cell activation. And something we thought about was beforehand was that we predicted sort of the role of phospholipid scramblase in those processes, and we thought about how it's more than likely something that's crucial for apoptosis to occur efficiently. And So in the study, there are like 6 or so parts to it, and they all investigated how protein kinase C delta affects phospholipid scramblase activity, and it found that scramblase activity is actually not cell type or stimulus dependent, and it also found that translocation and activation of the protein kinase C by something called FMLP, which is a microphage activator, the stimulation of it is actually transient and proceeds before scramblase activity actually ends up happening. And then we also saw that the protein kinase C is partly responsible for the activation of scramblase activity during apoptosis. And in the last section of the study, what they did was they had CHO cells, which actually lack both protein kinase C delta and scramblase, and they co-transfected them with either just the protein kinase C, just scramblase, or both together. And what they found was that actually scramblase activity increased dramatically if they had both the protein kinase C delta and the phospholipid scramblase in the cell together. And they measured that through the uptake of CPAF, which is a lipid. And they also found that if it was just one or the other in the cell, there was little to no activity of phospholipid scramblase, which sort of suggests that protein kinase C is necessary to help activate scramblase and apoptosis and cell activation.
Okay, so I'm going to talk about 2 different families that phospholipid scramblases. The first one is the PLSCR family, which is a family of 5 homologous phospholipid scramblases found in humans. And they are typically clustered on the Q arm of chromosome 3, depending on the like the 1 through 5 depends on like the specifics, of course. And then they all have a proline-rich N-terminus, and they also have a like motif area as well, except for the PLCR2. The motif areas will come into play later when I show the protein. And then they also have cysteine-rich regions as well, which is important for calcium binding. So this just shows a structure of PLCR1, which is 1Y2A on PDP, and it just shows the structure of the protein. So the PLCR1 is a family of the PLCR family proteins, and they basically are formed and anchored to the bilayer through a process called palmitylation, and that's just a process where there's fatty acid covalent bonding, and that's how they anchor, usually to cysteine residues on the membrane. And their cysteine-rich sequence is also required for this to occur, which is important for these proteins to have that there. And then they also have a protein kinase C phosphorylation site, which is important for important binding. And then these 2 motifs here, the PXXP and the PPXY, are also for important signaling. And then they're also involved with cytokine activity, and that was actually found through research which established them in the nucleus. And they're not quite sure of why this occurs or what the pathway is. It's still trying to be figured out, but it is what occurs. And this just shows the same protein, the PLCR1, with certain highlighted regions. So we have the orange region, which shows the proline-rich region right there. The red is the SH3 binding domain, which is also where the PKC is located, which is involved with Wnt signaling and other types of important signaling as well. And then you also have the cysteine-rich region, and then in the blue, and then the yellow is actually the single pass, which is the helix that is anchored into the bilayer. So they also contain a nuclear localization sequence, and that's a hydrophobic amino acid sequence, and they are important to important binding. Basically, it's required in order for the important molecule to bind to the molecule. The protein also contains 2 sites that have been identified, and then it's the major and the minor NLS site, and then those are just the base pairs that the first one's the major and the second one's the minor. And this just depicts in the red the 2 NLS sequence sites. The one on the left is the major and the one on the right is the minor. This shows a linear depiction of basically some of the binding sequences and areas. And so it just shows the IBB, which is an important beta binding area of the sequence. You have important subunit alpha 1, which is also involved with important binding. A lot of these sequences actually overlap each other, so the IBB and the important subunit alpha 1 through 4 actually overlap the nuclear localization sequence. And then the other family is the TMEM16 family. There's 10 members of this family, and they're in a lot of different types of organisms, from mice to fruit flies. The one I'll be talking about today is found into the Nectaria fungi. And they basically are transmembrane proteins. Depending on what type of protein in the family, they have a certain amount of transmembrane helices. This is a form of the 4WIT protein, which is a crystallized structure of TMEM16F, and it just shows the structure. As I mentioned before, they are transmembrane proteins, so transmembrane proteins have transmembrane passes where they loop through the membrane. The protein that I just showed has 8 transmembrane helices involved, and the protein is actually embedded into the ER, and it's where it's typically found. They have 59% helices, and they have they contain some beta sheets. And then their N-terminus and C-terminus region is also embedded into the ER membrane. And this shows some of the transmembrane areas, so like the inside, the outside, and the transmembrane helice passes that are highlighted. So we have the blue is the outside, the red is the inside, and the pink is the transmembrane helices that are the hydrophobic regions. This is from THMM, which is a bioanalytic software that shows the passes of transmembrane helices through the bilayer. And then this just depicts the sequence that I put into the program and base pairs where these specific components of the transmembrane passes are located. And then they also contain calcium binding regions and chlorine ion channels, which is important for enzymatic catalysis. So the binding site is located in a hydrophobic cavity in the membrane. And then there's a pore loop region that is located between the 5th and the 6th part of the transmembrane helix that is where the chloride ion channel is actually located. So this just shows the structure that in the blue is the calcium binding area of the protein, the TMEM16F protein crystallized structure. And that's just the highlighted calcium binding areas and the chloride ion channel areas as well.
We need that.
See how long it was.
Hopefully it's not too long.